In this video, we will talk about enzymes and how they help the body do reactions that may otherwise take years or even millenniums to actually go into completion. Okay, so first of all, we have to say what what exactly is an enzyme, right? So an enzyme is just a bunch of proteins with some RNA. Okay, so they're just proteins with RNA. Okay, and what makes enzymes special is that they actually can increase the rate of reactions, and when they do that, they're not consumed in the process. So what I mean by that is you can use an enzyme to do reaction A, and you get the product B, but along with the product, you also get the enzyme back. Okay, so in the same way, it's like drilling a hole in a quarter and putting some string in the quarter and using that quarter to get some soda from a vending machine, right? Back in like the 90s. You do the reaction with the enzyme, the coin, the quarter, and you get your product, the soda. And you also get your enzyme back, in this case, the quarter. So this is done legally though, and it's not as fun, but yeah. So let's just do that. Does reaction, and is not used up. Okay, now they can also speed up reactions, so they speed up reactions. Okay, so do you remember our carbonic anhydrase, right? So an example of this would be carbonic anhydrase. And here's a pro tip. Whenever, well, it should be like a review for you guys, but whenever you see ACE right here, ASE, that's just a suffix for enzymes. Okay, so if you have something like called proteinase, that's going to be enzyme. If you have anhydrase, that's an enzyme. Okay, lysase is an enzyme, so this right here would have to be enzyme. Okay, so if you remember, carbonic anhydrase is one of the kind of like enzymes that allow the essentially carbonic acid to diffuse, or not to diffuse, but to break up, dissociate into acids and bicarbonate, and that can be used as a buffer system within the blood, right? So carbonic anhydrase allows the buffer system to even occur. If you didn't have carbonic anhydrase, well, the reaction would occur very, very, very slowly, and this actually increases the the rate by 10 to the 7th. Okay, so that's a lot. It's going to be incredibly faster with an enzyme. Some enzymes actually will be so needed that without the enzyme, you may as well not even have a reaction. For instance, some reactions might take longer than the Earth has even been here to be completed, and with an enzyme, it could be done in like 2 minutes, maybe 10 seconds, right? So enzymes are needed within a lot of our biological reactions. Okay, so they actually increase the process. Okay, now one thing is that, yeah, they speed up the reaction, but one thing that they do not do is they don't change the equilibrium constant for the reaction. Okay, so they don't change the equilibrium constant in a reaction. Okay, that's nice, but what does that mean for us? Well, in simpler terms, it means that, well, if it speeds up the product formation, well, it's not going to slow down the reactant depletion, right? So if we have A going to B, right, and let's have a enzyme, let's call that E, right, for enzyme. Well, when B goes up, right, A is not going to go down. Like, what? Well, how should I say? the rate at which they change, okay? The rate at which B increases is going to be equal to the rate at which A depletes. So if we create two products, we're going to use up two reactants, right? So what enzymes do not do is that they don't give you three products and only use one reactant, okay? They're not going to change how many products you make or how many reactants you use. They're just going to speed up the reaction overall. And so the formation of B is going to show up equally to the depletion of A, okay? They're going to be at the same speed. So enzymes operate at the same speed. Operates both forward and reverse reactions at the same speed. Same speed. Okay. There we go. They also don't change the standard free energy within a reaction. So they do not change. They don't change the free energy. And if you remember from chemistry 1 and maybe 2, yeah, definitely 2, standard free energy is denoted as delta G, whereas this is Gibbs free energy. Okay, Gibbs free energy. Right there. Okay, so essentially, Gibbs free energy is essentially the energy created or consumed in a reaction. All right. So essentially, they're not going to increase the amount that is given off of a reaction. So if a reaction gives off 2 calories of heat, enzymes are not going to give off 5 calories of heat. They're not going to make it give off 1 calorie of heat. The reaction will always give out 1 or 2 calories of heat. Sorry. So enzymes do not change the amount of energy in the reaction. They don't increase the energy, they don't decrease the energy. They just increase the efficiency, right? Since we're talking about delta G, we'll just go over it, right? So if delta G, if Gibbs free energy is negative, then we can say that the reaction is going to be spontaneous. So reaction is going to be Rxn, okay? Is spontaneous. Whoa. So what does spontaneous mean? Well, normally we say spontaneous means, oh, it's just going to happen suddenly without you knowing. No. In chemistry, spontaneous means that it's going to happen to completion. It's going to want to happen, okay? So, for instance, in carbonic anhydrase, that reaction, that enzyme allows the buffer system to complete spontaneously. It allows it to go into completion. Without carbonic anhydrase, that reaction would not be spontaneous. It would not want to go to completion. Okay, it would take a longer time to get there. Like, the reaction is still going to happen, but it could take years, decades, centuries to happen. But if it's spontaneous, it's going to happen quicker. It's going to want to undergo the reaction. Okay, so what happens when it's not spontaneous? Well, when delta G is positive, the reaction is non-spontaneous. So you can consider the reaction not going to completion. It's not going to happen, right? So, for instance, if you wanted to convert, I don't know, oxygen into copper, just like some weird idea, is that reaction going to be spontaneous? No, it's never going to happen, right? The free energy is not equal, and it's not going to occur. That's just a dumb example, but that's the idea, okay? So if you wanted to put into thermodynamics, you would say that delta G is positive for that reaction. So there you go. But essentially, enzymes, they do decrease the initial energy needed to begin a reaction, okay? So let's say that it costs $10 to go on a ride, a roller coaster at Six Flags, okay? They charge an arm and a leg for that. My gosh, seriously. Enzymes are basically the friend that you have with you who's rich, okay? And they're like, you know what, I'll pay $8 and you just pay $2, okay? So the enzyme, your friend, allows you to go on the ride, the reaction, for a lower amount of money, energy, okay? They don't change anything else, they just change the initial energy needed to do a reaction. Sometimes you want to do a reaction, right? But you don't have enough energy to do it. And enzymes are like a little pick-me-up that allow you to do the reaction in the first place, okay? And whenever that happens, it actually speeds up the reaction, okay? Because enzymes lower the activation energy. The activation energy, okay? And they, essentially, they increase the rate constant and increase the rate constant. Okay, so in this video, K is going to denote the rate constant, right? So that's essentially how fast the reaction goes. Does it take 10 seconds, 10 minutes, 10 years, right? 10 musical albums. Music albums are like 40 minutes each. So that's, a legitimate measurement of time. So in summary, enzymes lower the activation energy, essentially the amount of energy to do a reaction, and when they do that, they actually increase the rate constant K. So that's how fast the reaction takes to go to completion. So now we're going to be talking about some diagrams. So these are actually called free energy diagrams, and they just represent the reaction's energy with an enzyme and without an enzyme. So for this case, the blue one represents a reaction without the enzyme. Now notice that over here we have some energy, right? So let's call this delta G, okay? Okay, well, this right here is the initial energy. And notice that without the enzyme, we're going to have to push a lot harder to go up this hill. Now, if you're an imagery person, imagine that you're that guy or person who has to kind of push this huge triangle. He has to push this huge triangle against this hill, right? So what do you rather do? Do you want to push that triangle up this huge hill, okay? Or do you want to push it up this hill, right? You want to push it on the pink hill. Why? Well, you're going to go to the same place. You're going from point A to point B, but with the pink hill, which is the catalyzed reaction, you're going to use less energy, and you're going to get a good product in the end, okay? You don't have to fight and tug this triangle up a huge transition state, okay, using so much energy to go to the same place, right? That's not very efficient. And so what enzymes do is that they allow the curve, the transition state, to be lowered. So essentially, the activation energy is supplied, okay? So they actually lower the activation energy, okay? So we can say, that this right here, this whole hill is called the activation energy. And so that means that you need a certain amount of energy to overcome that hill. Let's say 5,000 calories. All right, that's just a random number. But for this pink one, for this little baby hill right here, you only need about 2 calories. And so what am I going to pick? The 5,000 calories or 2 calories? I'm going to pick 2 calories. Okay. So enzymes allow you to lower the activation energy and therefore speed up the reaction. Now you don't have to spend 3 years climbing this hill when you could climb this other hill for like 2 seconds, okay? So now that we're talking about transition states, what exactly are they? Well, transition states are when bonds are broken and formed, okay? And you end up with the intermediate thing, Okay, it's not a product and it's not a reactant. It's weird. And it also has the option to become a product, and it can have the option to become the reactant. So you're stuck in between this area where you could become anything you want. You could become the product or you could become the reactant. Okay? And it is in this transition state where enzymes kind of efficiently place their hold on the substrate. Enzymes finalize their connections with the substrate. Okay, so I think that's pretty cool. And another important term that you need to know is that when you are in the transition state, you actually have binding energy. And so we're going to call delta GB binding energy. Okay, and this is really important in the transition state. And what binding energy is essentially multiple weak bonds. Lots of weak bonds between what? Between the enzyme, let's call that ENZ, and the substrate. Okay, now why does this happen? Well, the enzyme wants to form multiple weak bonds with the substrate in order to make it more stable, make it more what? More favorable to occur, right? So if we have an intermediate thing, okay, that is not stable, do we want to make it? No, you don't want it, right? You don't want to make a intermediate in the lab that gets on fire when exposed to oxygen, right? You don't want to do that. You want to make an intermediate that is very stable and not harmful to anything. And so the enzyme will form multiple weak bonds with the substrate in order to make sure that the intermediate is not harmful and does not break down easily and does not affect the overall reaction, okay? And those multiple weak bonds, we call that the binding energy, okay? So that is delta GB. So that's some vocabulary words for you to know. Now, going back to the structures, you have these little hills right here, right? And what these little hills are called, those are called transition states. So these are called transition states right here. Let's call that transition. Let's do that in black. Transition states. Right. Now you're probably wondering, okay, well, there are a bunch of hills. Isn't that bad? Don't we have to use more energy to climb up each individual hill? No, you don't. Okay? Again, let's say that the activation energy for EA, for the red hill, is about 10,000 calories. Okay? Let's say 10,000 kcal, right? But then the total sum for these would have to be like 1 calorie. Okay? It's not even 1,000 calories, it's just 1. If you want to think about time, the time it takes to climb up that red hill is about 34 years, and then the time that takes for the green hills is about 2 seconds. So even though you have more hills to climb, those are very small hills, and the enzyme allows you to do the reaction at a faster rate, okay? So again, they lower the activation energy, EA, and they actually increase the rate constant, K, for the reaction, okay? And if you want to know if this is favorable, just know that the products, the delta G for the products is actually lower than the delta G for the reactants. And so whenever that happens, this is going to be a negative. You actually lost, okay, some energy, which is good. So this is negative, and this would have to be positive. This is going to be spontaneous, right here, which means that it wants to go to completion. It wants to actually do this reaction. But how does the enzyme even lower the activation energy? You're just telling me what it does, but how does it do that? Well, the enzyme grabs the substrate in a chokehold or something, right? Very scary. And it holds it in a position where it will be more efficient to react with its surroundings, okay? So again, the enzyme holds the substrate in a position that allows the substrate to react more efficiently with its surroundings. Okay? Seems pretty simple. But that's a different topic which will be covered later. Oh, and the term for that is called catalysis. Okay, so catalysis enzymes hold substrates. Let's call that substrates right there. In a favorable position. Let's call that position in order to react. Okay, so an example would have to be, let's say you're trying to take off your shirt. Well, you're going to waste so much energy taking off that shirt that's really tight, okay? But then somebody, maybe a family member, helps you get the shirt off. And that is called catalysis. So that enzyme is holding you, your family member or something, is holding you in order for you to get into a better position so that you can take off the shirt, so that you can do the reaction efficiently, okay? So the enzyme holds the substrate in a favorable position in order for the substrate to do the reaction efficiently. Alright, now we're going to be talking about the enzymes and how the reactions occur, okay? So this is kind of like a nice series of pictures that actually show how enzymes move, okay? So right here, before we even begin, we have to talk about the enzyme. So this blue jelly bean looking thing right here is an enzyme, okay? So this is going to be an enzyme. Okay. Now what's this red thing in the jelly bean? Well, this one right here is called what? It's called the reaction site, okay? So this is called the reaction site right here. You'll see some textbooks refer to it as a catalytic site, which is a proper term. Okay, catalytic site right here is this red thing right there. Now, what is this mushroom-looking thing? Well, the mushroom-looking thing is called the substrate, okay? So that's called the substrate. Substrate. Now, essentially what happens here, well, first of all, we're going to do some shorthand. So E is going to be enzyme, S is going to be substrate, okay? So first of all, this enzyme is kind of molded, right? So it has this shape to it. And that shape is going to make the substrate groove into it. It's going to make that groove in the substrate hold, right? Again, this is catalysis. Right, so the enzyme is going to grab the substrate and hold it in a position where it's going to react efficiently, right? So the enzyme is connected to the substrate, and this forms the ES. So what is ES right here? Well, ES is a complex state, right? So this is the complex, ES complex, right? So ES complex. That's just essentially you're playing with Legos, okay? I couldn't afford Legos, so I never played with them. But let's say that you're one of the lucky people who get to play with Legos. And you have a red block and a blue block. You connect them together, you get the ES complex. They're not doing anything, they're just holding on to each other, okay? They're just holding on to each other. Now, over here, let's call this the ES, right here, the ES complex. Something inside of it switches, right? And so where they join right here, where they join is going to be very special. Because whenever a switch flips, they become the ES transition state. So this becomes the ES transition state. You're probably saying, oh, well, Brian, you know, that looks like the same thing. Why can't we say that the ES complex is just the ES transition state? No. In textbooks, you'll actually see this formula, right? So the ES does not equal the ES complex, or transition state, sorry. So ES asterisk just means transition state, okay? And that does not equal to the ES complex. Essentially what happens is that you connect your Legos and then you're going to allow them to activate. It's a bit weird, right? It's a bit weird. So let's use a remote controller, okay? So you have a remote, that's your substrate. You're gonna insert, no, sorry. You have a remote, that's gonna be your enzyme because it's gonna hold the battery, okay, in a favorable position so that the reaction will occur. So you have your remote, you have your battery, you put it in, that's gonna be the ES complex. Nothing else is happening, okay? But when you press a button on the remote turns on and activates a signal. That signal, or that process of actually turning on, is going to be called the ES transition state, okay? So just remember a remote controller with a battery. Together they don't do anything, but whenever you press a button, it turns them on and they become the transition state, okay? So again, this is going to be the ES transition state. We'll just call that asterisk, okay? And right here, they're actually going to make some product, okay? You'll see. So notice that this green mushroom converted to this weird thingy. It became beige. Well, what is that? You still have the enzyme, right? So we're going to call that E. But then we also have what? A product. So we have a product. And so really, that's just the enzyme and the product. So the enzyme and the product are going to disassociate to give you the enzyme plus the products, okay? So at one point, the product is still stuck in the enzyme, but then it's going to have to disassociate and rip off the product, okay? So overall, the reaction would have to be this, right? The reaction is this. The enzyme, and let's do that in a nicer color. The enzyme plus the substrate first gives you the ES complex. Okay, that's just a combination of the enzyme and the substrate, nothing else. Okay? But now the ES becomes the what? The ES transition state. And that is essentially the activation of the reaction within the ES complex, okay? So again, that does not equal ES. That is a different thing. So now the transition state, ES, makes the EP, which is just a combination of the product interacting with the enzyme. It's not reacting, okay? It's just being held in place. It needs to be let go, okay? And now the enzyme and the product are going to disassociate to give you back the enzyme plus the product, okay? Notice that you no longer have a substrate to work with, you just have a product and you also have the enzyme. So that shows you that the enzyme is given back. It is not used within, or I guess it is not used up in a reaction, okay? Let's zoom in on the enzyme. Okay, so this is what an enzyme looks like. It's very surprising. So imagine that you have a motif, or a motif, whatever you want to call it, of beta sheets, alpha helixes or helices, and some loops. Okay, so you have alpha helices, beta sheets, and loops, right? And so together they make a protein complex. Now, a very small part of this protein, okay, this whole array of proteins, has a little jelly bean in it. Okay, you can imagine that. Okay, so imagine you have a pack of Twizzlers and you take one Twizzler. That's going to be a fibrous protein. That's like a thread of protein. Okay, on that Twizzler, you see a little jelly bean on it. And that's your enzyme. Okay, that's actually your active site. So this right here, this jelly bean looking thing, is called the active site. I can't spell. So what exactly is an active site? So an active site is actually the platform in which the substrate is going to connect with and perform the reaction. So active sites actually do catalysis for the substrate, meaning they hold the substrate in a good position so they can react. So say they do catalysis. Okay, they do catalysis, and they're very small compared to the rest of the protein. They're small compared to the protein. Okay, there you go. Also, whenever they interact with the substrate, they're going to bond, but this bond is not covalent. It is a non-covalent bond. Okay, so the bond, let's say that you have your substrate, right, and it's going to be like this. So here's your substrate, it's going to interact. Yeah. So whenever it interacts, they have a non-covalent bond. Between the enzyme and the substrate. Okay. Now, what kind of bond is this? Well, it can be a hydrogen bond. So bonds can be hydrogen bonds. They can also be hydrophobic bonds. And they can also be kind of like ionic interactions, yeah. And actually, this would be the same thing. So you wouldn't have bonds right here. You would actually have interactions, okay? So you have interactions with this. Okay, there you go. So that is between the enzyme and the substrate, okay? Now, the active site can discriminate against substrates. So one active site is not going to go all willy-nilly to every substrate, right? So active sites are picky. They are so picky. Okay, it's like taking a kid to Denny's, right? They want some cake. They just ran out of cake and they're not going to eat anything else. They have to eat that cake and they're not going to eat anything else. So this active site can discriminate against multiple substrates. Only one substrate can fit into this active site. All right. So what happens if we want to interact with, I don't know, let's just make up something. It wants to interact with leucine or something, right? So if it wants to interact with leucine, it has two options. It has the option to interact with the L-leucine or the D-leucine, right? And depending on what it's made for, it's going to reject either the L or the D. It doesn't matter that they're the same species. What matters is the configuration. So it can actually be choosy, be picky over L or D, okay? Right? So they're very picky. They're made specifically for one substrate, right? And for that one substrate, the configuration for it also matters. If it's made for the left configuration, it's not going to go for the D configuration, right? So that's just very specific active site information. Also, let's talk about binding energy. So the interactions right here, right? So the H bonding, the hydrophobic interactions, and the ionic interactions, they all contribute to what? The binding energy. So essentially, delta GB is equal to all this, okay? It's equal to all the hydrogen bonds, all the hydrophobic interactions, all the ionic interactions, right? Within the enzyme and the substrate complex, okay? So that complex is going to start forming multiple weak bonds, but it's trying to do that in order to make the structure more stable, right? So we're going to end up with a nicer transition state intermediate, okay? If we didn't have those bonds, you would have a very, very unstable intermediate. Before I wrap up and go take a nap, I have to talk about the two models for enzymes and substrates, okay? So whenever an enzyme connects to its proper substrate, it has two ways to do this, right? So either the enzyme is going to have a lock and key configuration, so we'll call this lock and key configuration, let's call that config, or it's going to have the hand in glove configuration. Why is that? Okay. Well, this one right here, this enzyme is going to have a very firm, very rigid active site. Okay. It's going to always be in that shape, and that shape is very stereo-specific. So stereo-specific active site. Okay. Very specific active site. Now this substrate is specifically made for this enzyme, okay? They're going to have the exact same shape. So this is going to be the key, and this is going to be the lock, okay? For every lock, there is a key, right? That's very philosophical, but it's true. So this key, the substrate, is going to enter the very specific lock enzyme and form the ES complex, okay? So that's just them joining together. And of course, whenever they try to join, there's a bunch of what? Non-covalent interactions, okay? Just remember that. That makes the ES binding energy, right? So that right there, whenever they join, is very specific. This is firm, and this is going to retain its shape, okay? So everything has its same shape. It's going to keep being, rigid. It's weird. Okay, but over here in the hand and glove configuration, you notice that this enzyme has this weird blobby looking thing, okay? Well, imagine a glove. A glove, whenever you take it off the box, it has kind of the shape of a hand, but it could be folded and flexible. It could be stretched and retracted, right? So the enzyme, in the same idea of a glove, the active site is very flexible. Okay, so active site is flexible. Okay. Yeah, it has a weird shape, but whenever the substrate comes into contact with the enzyme, the blob kind of conforms to the enzyme, or to the substrate, okay? So imagine your hand, which has a very firm and rigid structure, I hope. It goes into the enzyme, which is soft. And this is going to be the glove right here. So this is the glove, right? And so whenever the substrate goes into the enzyme, it gives the active site its shape, okay? It gives the active site its shape. Okay, but you're probably wondering, oh, well, if it can be flexible, well, does that mean that it will accept any L amino acid or D amino acid? No, sadly not, right? So it's kind of weird, but it's still going to want to accept the original substrate, okay? Regardless, well, not regardless, but bottom line is that the enzyme is still going to check for the conformation, okay? Yes, it could be more flexible, but it's not promiscuous, I guess, right? So hopefully this video helped you out to understand enzymes. Next, we will be covering enzyme kinetics, so how fast enzymes react, how they bind, and whatnot. That's always a very difficult topic to cover, so make sure you do assigned readings and to do your practice problems. All right? So I hope you have a great day and that you continue to do well on your courses. So thank you and have a good day. Love you. Goodbye.